everyone. Thank you for joining us today for another Sleep Science Friday. My name is Isabella Santos Valentin, and I'm a PhD student at the Leibniz Institute on Aging in Vienna, Germany. And today I'm talking with Professor Vladyslav Vyazovskiy about his research and path within sleep science. Dr. Vyazovskiy is an associate professor of neuroscience at the Sleep and Circadian Neuroscience Institute in Oxford University, the PI of the Sleep, Brain and Behavior Laboratory, and also a member of the ESRS Scientific Committee. He did his PhD at the University of Zurich and postdoc at the University of Wisconsin-Madison. His research focused on understanding the spatial temporal dynamics of brain activity during sleep to uncover not only what sleep is, but also exactly why this phenomenon is so important. He's the recipient of many important grants, such as the Medical Research Council, Novo Nordisk, Marie Curie, and Royal Society. So thank you very much for joining us today, Dr. Vyazovskiy. And you are from Ukraine and did your university studies there, and then moved abroad to the University of Zurich for your PhD. I'm Brazilian, and also exactly today, 4 years ago, I moved to Germany to do my PhD here. And it has been quite an exciting road with some challenges of being abroad. For you, what were your motivations to do the same for your PhD, and how was your path from Ukraine to Switzerland? And how was your experience as a PhD student? There were some challenges that you faced that you still remember, and what you would advise undergrad students to take such a chance in moving outside their home country?
Yeah, thank you, Isabella. Thank you for inviting me to talk about my experience. It's really a real pleasure. And accidentally, it is now, I think, 7 years since I moved to Oxford, if I remember correctly. So I moved to Oxford on the 1st of December in 2013, right? So it makes it 7 years. So I also have a little anniversary these days, which of course was an exciting move. And it was the actually like third time I moved, because first indeed I went from Ukraine, from Kharkov to Switzerland. Then from Switzerland, I moved to the United States. Indeed, from the United States, I moved to the UK. And first I started at the University of Surrey in Guildford. And yes, that was my fourth move, because then I moved from Guildford to Oxford. So which, of course, when you move between cities, and especially between countries, it is a major challenge, but I think it is very stimulating. I definitely recommend, like, going to the kind of last part of your question, I would definitely recommend students to explore the world. So explore the opportunities, explore challenges, because this is how you grow, how you become resilient, how you network and make new friends, colleagues, and collaborators. So it's a very important experience. So to go back to kind of my story, so indeed I'm from Ukraine, from Kharkov. So it is the second largest city in Ukraine. And I studied at the University of Kharkov, and I always loved animals. So I wanted to be a zoologist. So yeah, so this was really my passion. My house was always full of animals, of pets of all kinds. I worked in a zoo in the summer. So this was really, I thought of myself as doing like something to do with animals. But of course, in practical terms, it was not that easy, especially we are talking about 1990s. So it was not a very easy time in Ukraine when the Soviet Union just collapsed. So the economy was struggling. There was not much resources around. Many people were leaving the country, and in fact, I was resistant. So I never thought that I would leave the country. So I thought that I would stay, and indeed I tried. So I started a PhD in Ukraine at the Institute of Cryobiology and Cryomedicine. But I quit after about a year. And I started looking around because it was just not progressing well. There was not enough resources, and although I really loved it, so my supervisor was a very, very talented neurobiologist, so we had great discussions. had great ideas, but it was just difficult to do research. And I started to look around, and I sent a few letters, and one of the letters I sent to Tom de Boer, who was a postdoc at the time at the University of Zurich. And this is something that really changed my life in a way, because he didn't just thrown away my letter, but he actually replied. So he replied. There was
Feels
no
awesome, no?
Yeah, so he sent me a printed letter, which was very kind. He sent me his PhD thesis, and for me it was really motivating and inspirational, so that someone actually takes time and replies. So I recommend now to all PIs to take seriously when you receive an email from a young student, especially from far away, who might have no experience of how to approach more senior colleagues. So do take time and reply, because this is how you can really make a difference. So Tom made a diffcast with Vyazovskiy Bobler and Alex Borbey, and they invited me for an interview, and then they invited me to stay for a few months in the lab, and eventually they made my PhD possible. So I started my PhD under supervision of Irene Tobler in Zurich. So
Wow.
I stayed.
I imagine that this is how you enter into the sleep field then.
Yeah, exactly. Yeah, although interestingly, originally I was interested in effects of cold temperatures on the brain and physiology. So, so in the institute where I started my PhD was actually about effects of hypothermia on the brain. And this is how I found Tom de Boer, because he worked with Irene on a very interesting model of daily torpor in Hungarian hamsters. So I found his work because of work on hibernation and torpor. This was my initial interest. And then when I came to Zurich, and then I found myself in one of the leading sleep labs in the world. So, and of course, this was a very interesting environment. So I was exposed to some very interesting work, very interesting ideas. And only my very first paper was on hamsters. And then I switched completely. So I started working on sleep questions. And only recently, only a few years ago, I came back to that. So now I'm working again, among other things, on torpor, on the state of hypometabolism, and I'm also breeding hamsters. So we have a hamster colony in my lab here in Oxford.
Wow. Nice.
It came the full circle, and it's really exciting.
Super. To go back to the beginning, to torpor.
Yeah, exactly. Yes. And to finish on your question, so you also mentioned, yes, that after Zurich, I then went to the US. And we can talk about this separate, it's a long topic by itself. And but after spending some time in the US, yeah, I felt that I want another change. So I'm up to another change. So this is how I came back to Europe.
Yeah, amazing. So actually now go back then to US. I'd like to know a bit more about your experience as a postdoc. So you did your postdoc in Chiara's and Julius Tononi's lab at the University of Wisconsin-Madison, which are among one of the most relevant sleep labs in the world. And particularly, I have always been a big fan of Chiara's work since back 2010, when I was still an undergrad student, when I read in one of her papers that we share with several other animals, and especially with Drosophila, the majority of the encoding network to control sleep. And I indeed remember how mind-blowing and intriguing this was for me, because I never thought about it, and how awesome would it be to study this further and further, not only addressing humans, but of course also in other animals, how sleep is so important and how it happens in our brains. So you were for many years in their lab and did your postdoc there, as I mentioned. So can you tell us a bit more about your experience there?
Yeah, absolutely. It may be better to start with, again, going back to Zurich, because as I mentioned, it was quite a big change. I feel that in general I'm really the luckiest person in the world, because I really had this opportunity to work first at the University of Zurich with Irene Tobler and Alex Borbey, and very closely with Peter Achermann and Tom de Boer in this laboratory, which was so stimulating, and they were really the best supervisors I can think of. So, and then after that, I went to the US. So US experience is quite important, and probably others may have different experience. So for me at the beginning, it was very challenging. So then, and this was really the best learning experience you can imagine, because Julia and Chiara, at least with respect to the work I was doing, they were more hands-off. So I was faced basically with the challenge to build the setup and to plan experiments and deal with practical things independently. So, and this was quite different from Zurich, where everything was really well organized. It was really so well organized that you really spend a lot of time just running experiments and analyzing results. So I had at least like 5 papers from my PhD only.
Wow.
These were different types. You know, you can have a paper with just 2 figures with 1 panel each, and now you have like 8 figures on the entire alphabet, like from A to Z.
Exactly.
Different scale. And then, yeah, when I came to Zurich, to Madison, Wisconsin, I came in the middle of the winter, and this is Midwest. It was cold. And I realized that I no longer can tolerate it. So I spent a few first months like in a state of hibernation, just trying to figure out.
Maybe with some torpor.
Definitely, yes. It was actually quite depressing. I had to learn how to drive, because you cannot live there without a car. And then I never drove in my life, so I had to learn how to drive. And yeah, and then of course you learn your ways, you build the setup. You know, the atmosphere in that lab was absolutely amazing. So it's this, you know, this very intellectually challenging, very intellectually rich. So you really, some of my best scientific discussions I had ever was with Julia and Chiara. So they were bursting with ideas, and this was really what kept me going. So and so eventually, you know, when I was building my setup, and at the beginning there was not much funding in that lab, so I bought my first grass stimulator on eBay for $45, and
wow.
And it didn't work, so I had to take it on a bus to the engineering department, and there was a guy who was like a genius, and he knew how to fix pretty much anything, and I brought him this machine, which was probably older than myself, and I said, can you fix it, because my entire postdoc depends on this machine. If you don't fix it, I'm done. And then he had used some magic, so it was screwdriver, and no, he basically fixed it. Then I brought it back, and I kept trying for some time, and then I got some advice from a few colleagues who were helping me a bit, like Marcello Massimini, who was a postdoc at the time. And I remember that day when I finally got my first evoked response. So I stimulated the brain of rats and would record the evoked response. And for a while, you know, I didn't know how it works. I would record just some artifacts. So there was like electrical artifact on a paper, and I would know how to interpret it. I would bring it to Julia, and I say, Julia, okay, this is what I got today. Okay, it's interesting. And then I okay, keep trying. Then I keep trying, and then next time I could literally like turn it around, bring the same, and say, okay, and now I have this. Because it looks meaningless to the same degree. and then, but then, yeah, I had an eureka moment how to do it right. So, and I recorded my first evoked responses. which was
Wow.
super exciting. And then really I started making progress. So I was
That
looking
sounds awesome.
yeah, it was, well, now it sounds awesome. Yeah, but then, yeah, we were really doing very interesting work, looking at how preceding sleep-wake history affects how the brain responds to stimuli. We use the walk responses as a proxy for excitability or synaptic strengths, and we obtained some interesting results. Yeah, and I spent 7 years in the US in the lab of Julia and Chiara. So the time really flies. So it was there was a really great group, fantastic colleagues. So other postdocs and PhD students. So Julia and Chiara were really good at finding the right sort of people, which is of course crucial for any lab, right? So the atmosphere in the lab is determined, of course, to some extent by the ideas you have by the supervisors, but also by the people. And it was really a great group. So we were good friends. We would spend a lot of time together outside of work. So this was great time.
Yeah, that really, now for sure it sounds awesome. And I can imagine that it was really a rich experience. And actually there and with them, you published a very important paper, which was the local sleep and awake rats, published in Nature in 2011. And as much as I can recall, this was your first Nature paper. And this study showed for the first time that after periods of sleep deprivation, some neurons in different cortical areas of rats' brain can unexpectedly go offline as if they would be under non-REM sleep, while some other neurons would be kept online. So this has, of course, a huge impact on the understanding of the effects of sleep deprivation, especially when we consider memory and learning abilities. And you already explained a bit, but deep to this study, how did you feel when discovering such an intriguing phenotype? Did you immediately think at first that this could be indeed the case? Or were you skeptical about it? And of course, later on, how did you feel to confirm your results and publish them in such an important journal?
Yeah, so I have to say that the idea behind this was not entirely new. So it was previously thought that sleep has properties of a local activity-dependent process. So it was especially kind of clearly articulated by James Kruger and Ferenc Obal in a series of papers they published, which suggest that sleep may be considered as a property of local neural networks, which was really a pioneering idea that kind of changed the way we look at sleep. So then, starting from early studies by Konstantin von Economo, the idea that dominated the field was that sleep is regulated by subcortical structures between the hypothalamus, basal forebrain, the brainstem, which basically decide when the brain would switch between sleep and wake. So this idea, of course, has a lot of empirical support. It's a very influential, very important idea. But the question was always, but how do those regions know that it is time to sleep? So where is this place where sleep need is accumulating, and in what form it is encoded, and how sleep process would start? So what would be the biological basis for that? And Kruger and Obal, they suggested that sleep is an activity-dependent process, and those neurons that were more active during wakefulness, they accumulate sleep need at that local level, and they may basically trigger the sleep process. So they did several interesting experiments. They were to test this idea. And then when I was still in Zurich, we did an experiment with unilateral whisker stimulation. So the experiment was very simple. We cut whiskers on one side, and then the rat was exploring the environment in the darkness using mostly half of the somatosensory cortex. And then we found that slow wave activity, which is an established measure of sleep intensity, was higher in the hemisphere contralateral to intact whiskers, suggesting that sleep intensity is not a global measure of sleep need, but it may be also expressed at the local level. And then when I was in Wisconsin, we took this at a new level. So because we established the technique of continuous monitoring of neuronal activity, so we could record spiking activity of single neurons in the brain, and this of course opened new opportunities. And it was actually remarkable that we could just see with the naked eye. So when you look at your recording, you could see an animal which is awake, moving. You could see very brief, what we call off states and slow waves, which were very much like localized. So just looking at the data is really the most important thing I can recommend. So look at the data, because this is the only way how to understand the data. So you may get an idea, new ideas based on that, and it's really important not to take a shortcut and start analyzing. You know, you can use a very sophisticated ways of analyzing, process the data, but this is not the way how I would recommend. So you look at the data, and this is how we observe this. And then, yeah, it took us a couple of years to really develop this paper and all the analysis. And we submitted the paper as a letters to Nature. And then I remember Julia comes to me. No, I think this was probably after we already submitted the revision. We got positive comments. And then Julia came to my desk and said, "We have a problem." I said, "What kind of problem?" He said, "You know, the editor wants us to rewrite the paper, so it is a full article."
Wow.
So we were even upgraded. So instead of like short letters, so we were asked, so it was considered an important finding, and everybody loves local sleep. So this allowed us, of course, to add more references and write a bit more. And yeah, so this is, and since then, you know, I think it is, of course, important. This idea is around for some time now, local sleep, which may indeed explain why we feel tired or sleepy. I think it is, though, and it changed our view, as I said, how we look at sleep. But it is time to start thinking, okay, what does it mean and how to move forward? So this is the interesting time right now to understand the biological meaning of that pattern.
Yeah, that was indeed an amazing paper. And actually now talking a bit about myself, one of my dream experiences is really to record live neuronal activity and see, as you just mentioned, a neuron spiking. And so I wanted to know a bit more how it was for you to get such training. Did you get this training in Karas and Julius' lab, or this was already done in Zurich? And to produce such an elegant approach for this paper, and of course for other papers that you published as well.
Yeah, this is a very important approach and method, because most of the conventional EEG, most of conventional sleep research is based on just EEG recording. So EEG in mice or rats is not very easy to interpret. So you can extract many useful metrics for that, but you still wouldn't really know exactly the origin of specific waveforms if you record just one electrode and depending how you reference it. So we wanted to go one step closer to the ground truth. So we want to know, okay, what the neurons are doing during sleep. So it was a bit challenging to set up the technique. So at the beginning, there was another DPhil student who was helping to set up the technique. So he had engineering background. There was another important collaborator in this project, Umberto Alcese, who is now an associate professor at the University of Amsterdam. He was doing his, I think, PhD at that time, and he was a very, very talented engineer, so he helped to set up the approach. So it was a bit scary to approach this area. So I started by like looking for all papers I could find. So I still have a folder from that time with maybe like 200 papers that I read. And you know, it is not like I learned everything, but it made me to be less scared of the technique and the novelty. So of course there were a lot of practical challenges that we had to face. But I remember when I implanted my electrodes and I saw my first neurons spiking, and I had a remote connection from my home, so I would even wake up in the middle of the night. I would go turn on the computer and look that the neurons, are they still there?
Oh, it must be really exciting to do this.
Yeah, it's exciting, and we are using very routinely this approach also in my lab here in Oxford, and the students and postdocs in my group, they really perfected this technique. So we are using more kind of refined ways to record neuronal activity with very kind of fine histology to confirm exactly where the electrodes are, have beautiful signals. So I think it's a very powerful technique.
Well, yeah, I hope one day I can do it. And so since 2015, just mentioned Oxford and your lab, you were an associate professor of neuroscience. How was your path from the postdoc until this point? And what were your biggest challenges as an early career researcher? And also on that, do you have any advice on how to navigate through these positions?
Mm-hmm. Yeah, as I said at the beginning, I feel I'm very lucky that I had good mentors and supervisors who were always and still are very supportive. These are people to whom I go for advice even now. And probably my first advice, again, going to the end of your question, is to well, I cannot say to choose the right supervisors, because there is a lot kind of under your control, but there is also some luck here. But it's really important to have good supervisors, good mentors who will help. And so I was helped, and this is really what helped me to navigate from being a postdoc to become a PI. I also would like to mention that before I ended up in Oxford, so I spent a couple of years at the University of Surrey in Guildford, where my closest colleague was Dirk Jan Dijk, which was also a very, very, very nice time, very interesting time. We also had very good scientific discussions, and Surrey, my short stay at Surrey helped me to move to my kind of more stable position here, because you know, when you change countries, you have to learn new things. And this was my first independent position. So I'm grateful to Surrey for actually helping me to learn how to become more independent. And there are no courses for PIs. That's the thing that it's crazy. Because yeah, at some point, okay, you decide, yeah, I'm ready for that. I'm ready to become independent, but you have no idea what you're talking about. Because this is a different world. It's a different world when you have to learn like a new way of behaving, new type of activities, how to deal with finances, how to write grants, how to write reports, how to supervise students. If you have teaching duties, there will be some teaching duties which can take a lot of your time and effort, administrative duties. So this is why you see more and more kind of a junior independent positions, like a fellowships, which help you to kind of progress further. But be prepared that it's not a very smooth process. Environment is key, is hugely important, that you have friendly, supportive environment, and count on your previous supervisors, because you will need the advice and support in going through these steps.
Sure.
Yeah, and maybe just last thing about that is I always recommend students in my group to network, to think about building a network early on. So you may think you're busy doing your experiments and then you don't go to conferences or you don't attend talks. You don't find time to meet with visitors or even talk to others in your department. That's wrong. That's fundamentally wrong. You may have beautiful nature paper then, but if you haven't developed your skills to communicate with people, and if you haven't met people who will be your future collaborators, colleagues, reviewers of your grants and fellowships, then you still have a long way to go. So this I definitely would recommend. And again, when I was in Zurich and in Wisconsin, I'm grateful to my supervisors that I had this opportunity. We would always go to conferences, and this helped me to make connections.
Yeah, for sure, it's one of the best opportunities to really talk to people. No?
Absolutely, yes.
So now comes the question that I would say that can also fit into all this new adventure as a PI and a professor. How do you balance your time among all these duties? And as a PI, what are your biggest goals for your working group right now?
My biggest goals?
Yes.
Yeah. I'm learning. I'm still learning, and probably I will always learn because you have new challenges. For example, my latest addition to my duties is that I joined one of the colleges here at Oxford. And I now have like a quite substantial teaching load in addition to everything else. So this last term was very, very challenging because everything else continues. So I still have a large group. I'm supervising several PhD students. There are postdocs in the group. So what helps is that the infrastructure is basically there and expertise is there. And there are several very experienced people, very motivated, very knowledgeable, who are helping to drive projects forward. So time management is hugely important, and also priorities. So it's really important to decide what is more important, what is less important. And now I basically learned to, at any given point, I know that there is a deadline tomorrow or in 3 hours.
Wow.
And yeah, it was like psychologically, it was very difficult because I hated to have something that is unfinished or not done. So, and then, you know, it does not get better, it gets only worse, right? So you have more and more things to do, and you eventually learn, kind of exist in this ocean of things you have to deal with. Maybe when you get older, you know, you start, you cannot think about everything at once. So maybe this is what, like, forgetfulness. It comes with your age, so you forget about everything except the most urgent thing. So, and then other things that you don't actively think about right now, they don't really exist, they don't bother because of that. And then you look at your to-do list, and then you feel really bad about things, and then forget again. Yeah. So this is the...
Okay. So now talking a bit about like lab work right now, one of your research interests is the relationship between sleep and neurodegenerative disorders, with a couple of papers published in the last years and this year as well. And your groups and others already demonstrated, using different animal models, the importance of sleep for the brain activity homeostasis. And it's clear that it changed with age, that the local sleep is also altered altogether with our sleep architecture as well, now talking exclusively about humans. Moreover, a few studies demonstrated that Alzheimer's disease patients show episodes of sleep deprivation and a syndrome called sundowning, which could be a misalignment of their circadian rhythm. In your opinion, this relationship is a chicken or the egg problem? Which one might come first? It's a lifestyle of our modern society which increase our sleep deprivation episodes that push the brain to be less and less healthy. Or could it be that it's a problem even earlier than sleep itself, but rather the circadian disruption that fails to orchestrate such behaviors as we grow older? And to this, what is the next big step in your view that could come up while studying sleep and such diseases?
Okay, so this is again many questions. So I appreciate that you mentioned circadian rhythms as a very important factor in these discussions. Here in Oxford, I have an absolutely amazing opportunity to work very closely with really like world experts in circadian rhythms, like Russell Foster and Stuart Pearson, Arti Jagannath, and a few other people who are leading researchers in the field of circadian neurobiology. And this is, I think, hugely important for the sleep field, for sleep scientists to talk more with circadian biologists and vice versa, because this is where you really find new ways of how to look at your data and how you to understand that. So there are several projects going on in my lab. So we are not only focusing on sleep and neurodegeneration. To me, sleep is such a complex phenomenon that for me it was always challenging to focus on just one aspect. So when you focus on one thing, you start thinking, no, but how come I'm not including this aspect, which is also important? This is probably my main challenge. I cannot decide. This is why we have like several kind of directions. One is about sleep and metabolism, another sleep and circadian rhythms, another like cortical versus subcortical sleep control, and then indeed another project on the link between sleep and cellular stress, which is considered to be one of the important factors which is implicated in the link between sleep and neurodegenerative disorders. So like Alzheimer's disease is clearly concerns problems with proteostasis. Mm-hmm. And we have some initial tentative data, mostly from UPenn, from Ginny Naidu lab, suggesting that wakefulness and sleep deprivation are associated with accumulation of ER stress and the plasmic reticulum stress. And we now have indeed a project to investigate this link, with Ginny being one of the collaborators on that. I think we need to be open-minded, a bit skeptical and critical, exactly when we look at the directionality of causality between sleep and neurodegenerative disorders. So we have some really striking examples in this area. For example, when we talk about RBD, REM behavior disorder, and Parkinson's disease, it is really well established that there is a connection. So so if you develop RBD, it is very likely that you will develop Parkinson's disease. So this is a very nice example how looking at some sleep mechanisms can inform understanding of neurodegenerative disorders. With Alzheimer's disease, I think more work needs to be done. So we do have some very interesting evidence that there may be a relationship, but right now I wouldn't want to say that yes, there is a causal relationship, and if you don't sleep enough, you will develop Alzheimer's disease. So I will not say this. And this comes to the indeed choice of methodology and the choice of experimental models. Right, so I think you mentioned that this malfunction of the circadian clock may contribute to age-related alterations in sleep, which may indeed predispose the individual to the development of neurodegenerative disorders. But then we need to understand indeed better why circadian rhythms weaken with age. And another is experimental models. So a lot of our work is about mice, and maybe we can talk also later about how different animals sleep. more naturalistic approaches to sleep studies. But mice, when they get older, they actually sleep more. They do not sleep less, like is typical for humans. So we need to be critical. So because it is not an inconvenience, but it's a potentially very useful and important information, which should inform our experiments to really close the gap between sleep disruption and neurodegeneration.
And for sure should be kept in mind, no, that even though the animal model can answer a lot, most of the time they are inside the lab environment, which of course is not the same as being in the wide open nature. We could also, of course, impact the behavior that is so closely related to the environment as sleep. So, of course, to make such a bold statement that one causes the other, it was more of a provocative question, because often I think a bit about this and how difficult it could be that one day one can answer this, if it's even possible.
Yeah, I so much agree with you about our overall approach, that we are trying to understand sleep in the conditions which are completely different from those where sleep evolved and the respective genes were selected for. Right, so imagine our laboratory mouse, our favorite model, and mice are probably they're happy the way we keep them in the lab. You have, imagine you have tons of food under your feet all the time. And you know exactly when it goes light on and off, and there are no predators around except humans who want to do something to you in an injection or something. But it's really so different from naturalistic conditions where animals have to balance their sleep need with all other important drives. I think so the idea I have is that there's no way we can understand sleep by looking at laboratory studies only. It is too bad that only a few labs now are actively looking at sleep in the wild, and they're making absolutely fascinating discoveries. Again, when I was in Zurich, Irene Tobler, she contributed this first initial observations of how different animals sleep. This phylogeny of sleep field really started back then, and then several labs picked this up, and fruit flies became a popular model. And then Niels Rattenborg, when he was in the laboratory of Rubenka, they started looking at how birds sleep during migration, and now Niels is doing really interesting work, and John Lesku with animals in the wild. So these are really hugely important studies that will provide important
Absolutely. And now taking a bit of this link, you commented in Current Biology a few years ago about studies that were trying to understand the influence of lunar phases upon human sleep, which back then made me think about one of my favorite paintings, which is The Sleeping Gypsy by Henri Rousseau, which depicts exactly this naturalistic point of view. Is the imminent danger of a lion approaching a sleeping woman under the moonlight. How much do you see the correlation between science and art in general? And going a bit further in this direction, do you think there are many fundamental, still open questions, perhaps about sleep, of course, perhaps hidden in art pieces?
Okay, that's a great question. Talking about the effect of lunar phases on sleep, so indeed, evidence accumulates that the moon is not just out there, but it has potentially interesting influence on sleep, and both in humans and in animals. Again, in the recent study, I believe from Peter Merlo and Niels Rattenborg, they looked at sleep in starlings, and there was definitely an influence of the moon on how much they sleep. And this is where you see that the environment plays a major role in sleep. So environment determines how you sleep. And this tells us something quite fundamental about what sleep is. If you can sleep, if you can decrease or increase the amount of your sleep depending on environmental conditions, So we need to reconsider our kind of very narrow view that sleep is for memory consolidation, let's say. Okay, because there is probably more interesting than that. Yeah, and yeah, you mentioned this painting, Manly Rousseau, which is fantastic. I love it. So I also saw it, the original in the New York Museum of Modern Art. I remember, and it nicely depicts again this influence of the environment on sleep. Sleep is about your relationship with the environment. It just cannot be defined without taking this into account. We talk about defining criteria for sleep. You don't move and you do not respond to stimuli. Your sense arousal threshold is elevated. These are two things how you interact with the environment. You get information from the environment and then you act upon it. So you respond to that. And both the things are diminished during sleep. So we absolutely must take into account this interaction between the organism and the environment when we try to understand sleep. And this is why, these pieces of art, the paintings, like you mentioned, they can stimulate some ideas of this sort. So art is indeed about imagination, and the difference between science and arts. Perhaps most fundamental is that science is based on your objective measurements of phenomena, while arts is about something more subjective, right? About something more having to do with imagination. But here where the two can actually come together. So if you look at a painting like we did right now when we are discussing the Sleeping Gypsy, so we do not actually know what Henri Rousseau had in his mind.
Absolutely. Yeah.
This is we are trying to interpret. This is our interpretation, which depends, of course, on our history of everything, like our knowledge, our ideas, our mood, maybe our time, the time of day when you look at the paintings. So it will affect our interpretation. And now when you think how we deal with scientific data, when they're just numbers, they're meaningless. They must be put in the context. They must be interpreted. And the imagination, it precedes knowledge, as Nobel laureate Richard Axel said, "Before you know, you must imagine." Which is a very nice quote. So you have these tables or graphs in front of you from your data, and then you try to make sense of it. And this is where you use your imagination to put the data in the context. And this is quite remarkably, is that depending on your experience, your knowledge, on which dogma you adhere to, you may interpret the data completely differently. And this is, I think, probably the biggest problem, right? Sometimes you read the paper and then you see how the authors conclude what they conclude from the findings, and you say, like, no way, it just doesn't mean that because of this and that. So I think this is when you were asking about the relationship between science and arts. I think they all converge onto kind of our subjective view on the topic and what we make of the painting or of the data.
For sure, and I think that this could be actually a very powerful source exactly to this point, to imagine. Because for sure, I looked to this painting already many times, and I guess each time I can think about something else and make exactly this story on my mind. What might he be thinking when he was painting this? What was really the goal to depict such a common behavior and such a widespread behavior that for sure it's everywhere, in all the days in our lives. Mm-hmm. So it was a really nice point of view. Now going a bit far-fetched than perhaps our last question, I was asked this question, and since then I'm always curious to ask this as well to the professors that I meet. if you would have no constraints, what would be your dream experiment?
Yeah, that's a good question. Yeah, I can think about quite a few. Um, um, well, probably my dream experiment would be to look at how animals sleep in the wild, those animals that were not studied yet in the wild and their naturalistic conditions, and to have and to do this experiment on a large scale. So when we look at sleep in any subjects, in animals or in humans, usually we have a small cohort which is studied in a very controlled conditions, and we always miss the big picture. Again, talking about the importance of the context, right? And if you think about sleep in a particular individual or sleep in a particular species, so the way that individual or the species sleep, I think, can only be understood in the context of everything else around, of what happens in the entire ecosystem, right? Because so you need to coordinate the timing of your sleep activities with everything else, and other animals also have to sleep. And we just don't have a methodology. We don't have a good approach to look at this landscape of sleepscape in the in the ecosystem, because I'm sure that this will provide some very important fundamental answers. So this will be my dream experiment.
Yeah, this sounds very interesting. And how do you think this is close to happen? Or have you thought about these possible methodologies that one could use to tackle and to this question and to do this approach?
Well, it is very difficult. Probably we need to start with some modeling studies. So we and the technologies they develop fast. So you probably can create an virtual ecosystem, or you can work with artificial life agents, whom I don't know. So you can create like a world, like a dream world, with a lot of species which exist in a certain trophic relationship with each other, and you can vary environmental factors. So and you could make some progress with that, but you cannot make sufficient progress unless you know how those animals sleep in the wild. So you still get back to the initial problem, but we can start with maybe some modeling approaches.
Yeah, for sure. And hopefully this is the future of sleep science.
Will be the future.
Yeah. So now, as a final remark and a final question, which advice would you give to students like me that are crossing the finishing line of their PhDs and about to enter this vast world of being a postdoc and a fully educated scientist, either in academia or even in the industry?
Mm-hmm. Yeah, you are approaching a very important milestone, of course. And the first thing I would say is, do not be shy and do not be scared to take a decision if you realize that this decision is wrong. Again, you know, there are many factors we just cannot take into account. But do not be afraid to take a decision. This is something that may block some of us, because there are so many opportunities, there are so many dangers that are real or perceived, which prevent you from making steps. So don't be afraid. So if you moved on, if you, let's say, started a postdoc, or you decided to go to a company, and then you realize that this is not for you, consider this as a very important learning experience. You know, you have time, so you have time. Just make most of that. Move on. You will. I don't know. A few years down the line, you will forget about that. So don't think only about the now, but think also about the future. So as I mentioned, planning is very important, and also networking is hugely important. Try to meet people, because you'll need letters of recommendation. So what happens sometimes, the students who are close to finishing and they start applying and they're asked 3 letters of reference, and you realize suddenly that you can ask your supervisor, but you don't really know any other senior people who knows you well enough to do that. I think this is something that you should not forget. And yeah, there is nothing wrong with leaving academia. So academia is not for everyone, and sometimes leaving academia is perceived as a failure, like you failed as a scientist. That's completely wrong. So I really would encourage everyone to look widely. Academia has its perks, so it is exciting. It's also very challenging. And you know, I don't want to say much about or the world outside of academia, because I don't have that experience, but certainly I wouldn't consider this as a failure if you decide not to stay in academia. This would be my kind of general advice.
Thank you. I should take this in account for sure. So thank you very much for joining us for this Sleep Science Friday. It was really nice for me to interview you. I hope you liked it as well.
I loved it.
Thank you.
Thank you very much, Isabella. It was a pleasure to talk.
Thank you.